So I am pleased to introduce our speaker today, Wendy Hodgson. Wendy is the Herbarium Curator Emerita and Senior Research Botanist, celebrating her 50th year working at the Desert Botanical Garden in Phoenix, Arizona. Her research focuses on the agave family, particularly the systematics and role of agaves, yuccas, and Hespero yucca in pre and post contact cultures. Wendy also focuses on the Southwest floristics with a special emphasis on the Grand Canyon, where for 35 years she has searched for plants. She has authored or co-authored 9 species new to science, including 3 from Grand Canyon. Wendy also focuses on the Saran Desert food plants, publishing Food Plants of the Saran Desert, winner of the Klinger Book Award presented by the Society for Ethnobotany. She's an advocate for collaboration, working closely with numerous federal agencies, conservation groups, academia, community scientists, and indigenous peoples in the study and conservation of plants and their habitat. Please join me in welcoming Wendy Hodgson, speaking with us today about the Grand Canyon, more than just stunning geology, vistas, and strange squirrels, her green heart, the unsung legacy of plants. Wendy, if you'd like to share your screen.
It should be shared. Is it up?
Yes.
Okay, good. Well, first, thank you so much, Elizabeth, and thank you all for inviting me to talk to such a great group. It really feels it's an honor for me to be here. Thank you. And it's great to see some of my friends here, too. So let's get right into it. So Grand Canyon is big. River miles is 277 miles long, and if you were walking from one end to the other, it'd be nearly 800 miles along the rims, going in and out of side canyons. It's up to 18 miles wide and a mile deep. It has over 1,800 plant species, nearly half of Arizona taxa. And this is significant because Arizona ranks 3rd or 4th in the country with total number of species. It is also a hotspot for endemic plants. We've recognized at least 15 endemics in Grand Canyon region, and most are arid adapted. And we have a lot of near endemics, mostly in the Grand Canyon, but with outlier populations. We have about 20, and again, they too are very well adapted to arid and semi-arid regions. So the question, why is there so much diversity? One reason, it's on the Colorado Plateau, a region that's known for high endemism and species diversity. It's a meeting place of 4 of the 5 deserts of North America. And if you ever walked or taken a burro down into the canyon, you experience a wide range of elevation. And with this too, you have a lot of different aspects and slopes. There's a lot of diverse geologic formations and soil types, like this little evening primrose that just loves this tough, powdery soil. And there's, of course, the Colorado River, but also the seeps, springs, and creeks. Seeps make up less than 2% of the canyon, but supports a very high diversity of not only plants and animals, especially insects. And the canyon has experienced extensive human occupation for many years. And of course, this influences plant distributions as well as speciation too. So one way, an important way to figure out, uncover the canyon's plant secrets is through the making of herbarium specimens. And that is something I do a lot of. Herbarium specimens help us understand what grows where and answer so many other questions now and in the future. So Grand Canyon National Park has an herbarium. It's dedicated to plants. It's the only one dedicated to plants of Grand Canyon National Park. And you can access the specimens through the southwestbiodiversity.org link. Now, unfortunately, researchers will overlook small herbaria, and this is unfortunate. This is ocotillo on the left here, and it's common in the western end of Grand Canyon. So what happens if a researcher doesn't look at herbarium specimens from Grand Canyon Herbarium? He published in Vascular Plants of Arizona the treatment for the ocotillo family, And if you notice, there are no dots for occurrences up in the Grand Canyon. So it's really important for researchers, especially in an area where there's such diversity of plants, to make sure they hit the herbaria that's specialized in them. Grand Canyon collecting is really fun. It's no walk in the park, so to speak. There are very few trails. There's many challenges: too little water, too much water, a lot of rattlesnakes, cliffs, exposure, lots of spiny plants. And the plants also grow on really sharp limestone, like mauve limestone. All these challenges. Well, despite all these challenges, there's a lot of people that did a lot of great collecting, dating back to the late 1800s. And if you notice, all those in yellow, those are women. Half of the people that have done so much collecting in the canyon were women and are women. These four remarkable women face these challenges: Dr. Elzada Clover and Lois Jotter from University of Michigan, Susan McKelvey, and Rose Collum. Not only did they collect regular plants, like herbaceous things, but they also focused on cacti, agaves, and yuccas that are really difficult to collect under the easiest and the best conditions. So Elzada Clover and Lois Jotter, they were the first women to successfully float from Green River down to Pierce Ferry, all the way down the Colorado River. But they were the first people to do systematic botanical exploration along the Colorado River. And with their collections, they named several new species new to science, including these cacti. They published in these publications, as well as in new species publications as well. And these two publications, the one on the left is by William Cook. It's hard to find, but it's a great little book talking about their expedition. And then recently, Melissa Savigny came out with Brave the Wild River, which is also a really good narrative with photos of their expedition. And both of them complement each other really well. Now, Susan Delano McKelvey, she was actually first cousin to Franklin Delano Roosevelt. She ended up at Arnold Arboretum. She was very interested in monocots, especially Lilium, and she did a monograph of the genus Lilium. She was also really interested in yuccas, and she would make trips out west. But what was interesting to me, too, that she was a botanical illustrator, and that is her illustration, pencil illustration on the right. So she made her trips out west, including Grand Canyon, but she stuck to the rims because she was driven around in this car by her chauffeur, Hamilton. So she was restricted pretty much to the rims. She did name and describe species from the Grand Canyon, including Hesperoyucca newberrii, which we'll talk about more later. And this is a species that I'm really interested in as well. Also the kaibab agave, Agave utahensis, subspecies kaibabensis, she named and described, new to science. And the third, or the fourth gal, Rose Collum, she was a charter member of the Desert Botanical Garden, and she was well known to a lot of scientists, including Britton and Rose of cacti fame. She collected a lot in the Grand Canyon. Several plants are named for her. She was finally, about 6 years ago, inducted into the Arizona Women's Hall of Fame. But importantly, she was the first botanist of Grand Canyon National Park in 1932. So to answer these questions, we want to find out what goes where. This is, of course, the foundation for conservation of species and habitats. And as you can see, we've added over the years quite a bit of taxa to the Grand Canyon's flora, as well as species new to science, including this little kaibab skullcap and these two manzelias, or stickle leaves. They're unusual in having white flowers. Manzelia wallapiensis, named for the Wallapai Nation, and of course Canyonensis for Grand Canyon. And they have interesting distributions too. Canyonensis occurring in the eastern half, and Wallapiensis in the western half. And this comes up later in our talk. There are several publications. The first checklist and only checklist was published in 1987. We have additions to the flora that are published. Of course, new species are published. And then these more popular books, a really good one, River and Desert Plants of the Grand Canyon, that Lori McCarrick was one of the three co-authors. It's a great book. But we need to do an update, at least with a checklist or an updated flora, which would be a lot of work, but it really needs to be done. So there are a lot of rare plants in Grand Canyon, and one of them that the park has spent a lot of time and effort and a lot of interest in is this little legume. It's called century milk vetch. It's Astragalus cremnophylex, cremnophylex. It's a tiny little thing. You can see the pen to give you an idea of the scale. It grows in these cracks of soil in the Kaibab limestone. The Kaibab limestone is the uppermost level in the canyon, within 25 feet of the rim, which I find interesting because it's been suggested that with upward warm drafts from the bottom of the canyon, it can influence plant distribution and plant species on the edge of rim. There are only 3 populations known, and so threats are, there's only 3 populations known. And so, of course, and there are other threats: drought, herbivory, limited seed dispersal, limited habitat, and of course, all this contributes towards reduced genetic diversity and vigor. Well, Fish and Wildlife Service developed a recovery plan to find 11 different populations, 8,000 plants in each one. And so the Park Service, Grand Canyon National Park, with volunteers, it was their charge to survey for more populations, as well as monitor them, map them, collect seeds. Now, one of the populations, big ones, was at Maricopa Point. Unfortunately, there was a parking lot there. So they took out the parking lot and they restored it, and they got rid of it, restored the area, and then they built a greenhouse. And in the greenhouse, they germinated a lot of seeds, and they planted out the plants to augment the populations. It's a very slow process, but it is gaining. We have positive gains on these on this species. There are many invasive introduced species, about 200, 11% in the flora. And one common one that people know about most is the tamarisk. Interesting, in 1938, Clover and Jotter, they recorded 17 non-natives and included tamarisk, and this is pre-dam. Glen Canyon Dam was developed 1963, 1964. But the tamarisk did reach the canyon in the late 1920s, and it dominated the corridor, the whole river corridor, after 1963. And it was in the 1980s that they think that the tamarisk was established in side canyons. So Laurie McCarrick had this incredible, huge funded project to focus on as many side canyons as possible to find their seed source and then go back up with saws and whatever and get rid of them. And I was part of it a little bit, and it's to this day, it amazes me how much work had been done with staff and volunteers. This was extremely difficult work. Look at the number of tamis removed from all those tributaries. And they also monitored any impact of other vegetation, wildlife, water, or soil in the area, because we used an herbicide. So their publications, Laurie was a co-author in them, and they found that there was little change in the native species richness pre and post removal. But they cautioned too, this was a short amount of time, and there were drought periods within this time. But they, as with every other study, we have to do further study, further research on this. But this study did, and this project did reinforce the importance of controlling invasive species before their establishment as soon as possible. One cool thing that came out of this too, it got us into areas that we never would have been able to. So we were able to collect and document plants with herbarium specimens from these. So nipping in the bud, so to speak, this is Sahara mustard, Brassica ternifortii. It's really common in the Sonoran Desert and Mojave deserts as well. In 1920, it was first found in the country in California. And I happened to collect the plant in 1999 at Lee's Ferry in Grand Canyon. And that appears to be the earliest documentation of the plant in the canyon. But as you can see, thousands of plants have germinated and removed over a relatively short amount of time. If you get good winter rains, this plant's gonna come back with a vengeance. I've been to Lee's Ferry recently, and I don't see a lot of the brassica turnifordii up there. So I think we made great gains because of past removal activities. So there's a lot of this project really interested me. It's restoration projects along the Colorado River. And I want to preface this too with saying that there's a lot of research going on under the Bureau of Reclamation involving USGS, Grand Canyon National Park, and so many stakeholders in monitoring, mitigating the effects of Glen Canyon Dam. And they're focused mainly along the corridor. This one struck my interest, and it's planting. We're not going to get rid of the tamis in the corridor, but if you do plant and establish cottonwoods and willows like Goodings and coyote willow, they are going to do really well in cutting back tamarisk establishment. So watering them, though, depends on backpackers or people going down the river and river trips. The one in the middle is a very old Goodings willow. It's at River Mile 209 on the Wallapai Nation. That was an old plant when John Wesley Powell went down the river in 1868. We knew it was dying, so cuttings were taken from it. They were rooted, and these saplings were grown, and so we wanted to take it. They wanted to take it back to that site and plant them. Well, it's 209 miles. You have to start from the beginning, at least ferry. And so we strapped those plants to the side of the rafts, going through all the rapids. And this is actually the boat that we tied them to. We did get them to the site. We dug these big holes, planted them, caged them so beavers didn't get them, and put a sign up, please water these plants if you're in this area. Unfortunately, we lost them. But there's been so many other success stories, but it would have been really cool. And who knows, we may try this again in the future. So one of the research aspects that we're interested in at the garden, as well as others, is how the canyon influences what grows where and how they evolve. So it takes a lot of time, as you know. It's getting out there, knowing what's normal and what's not normal. You pick up patterns in your observations, and of course, documenting them. You tie that in with all these other tools that we have in developing phylogenies, and also uncovering and just new, what we call a cryptic species with our work. Instead of one species, it may be two or more that has been overlooked. And so when you have these, recognize these cryptic species and you name and describe them, you increase probably the endemism, because probably they are only found in that area, but also you increase the diversity, and you keep plants from falling through the cracks when you do this kind of work and figuring out what exactly grows there. So two examples, cryptic species, involve thistles. And thistles are really sort of a pain to make specimens of too, especially when you have to get the roots. And a good friend of mine, Jen Ackerfield, who is the world expert on thistles, she always says, yeah, this'll be a mess. And indeed they are. So this is Buck Farm on the left, Buck Farm thistle. And it's up in Marble Canyon, and this thistle grows in these seeps. And they have big, thick leaves. And it does remind one of Rydbergii, which grows in southeastern Utah, northeastern Arizona. But to me, there's something off with it. There's something different. The molecular is suggesting it probably is Rydbergii, maybe, maybe not. On the right, we have Cliff Springs thistle. It's found off the north rim in one spot and then another population below it. And the molecular is indicating, well, that's probably Rydbergii too, but you can look at it, compare it to the one on the left, which is more like Rydbergii. They're two different things, very different. So I think there's some cryptic taxa here, as well as if you go downriver to Lava Falls, which is about River Mile 174, and then you pick up these. And these can grow up to 9 feet tall. So you imagine making a herbarium specimen of this with roots. And so we don't know what this one is either. It could be Mojavensis, Virginettes, we don't know. So it's really difficult to get to do molecular on this, but this is something that we're hoping to sort out with other researchers. So this little cutie, we call it the California bear paw poppy. And So Fish and Wildlife, headed up by Jesse Miller, were to determine if this species should be listed as threatened or endangered. And so, but then there's this population in Grand Canyon that Art Phillips thought was different from all these other ones. And he said, well, for one thing, they grow on a very different soil. They grow on Muav limestone instead of gypsum. And morphologically, they're different. They're taller plants. The capsules are bigger. And so Jesse put together a team, sort of big collaborative effort involving Fish and Wildlife, Grand Canyon, Wallapai Nation, because some of the plants grow on Wallapai Nation, USGS, University of Nevada, Las Vegas, and DBG to search for more populations and document them, revisit older documented populations, get more morphological and molecular data, and all so far it is supporting that this is indeed a different taxon, possibly subspecies. Well, this plant, by the way, is really hard to get to. So there's Jessie Miller on the left there. She's the queen of the spotting scopes. And in the middle, we look up there and she's zoned in on that little diamond up there. She sees a 2-foot tall Arctomicon up there from there. And so it's very rugged. This plant loves to grow in the most rugged parts of Grand Canyon imaginable. And so we're trying to decide, it's 1 p.m., should we go up there? We decided not. And this was after we were chased by bees. We decided not, so this will wait for another time. So studying agaves, nolinas, and cacti, those two are a lot of fun. They're difficult to make specimens of. And so DBG, with other collaborators, we are assessing the cacti and agave family for the IUCN Red List assessment. And especially for cacti too. And cacti are the 5th most endangered organism on the planet. And so to do these assessments, a lot of it's based on actual and estimated area of occurrence that are based on documented collections. Well, with cacti, yuccas, and agaves, there's just too few collections. And if there are collections, many have poor material or data. And so and it's involving very complex taxonomic groups too. So it makes it really hard. So we want to get the word out in how to make really good herbarium specimens of these difficult groups. So 81% of the agave family collections, herbarium collections in SignNet, are in Grand Canyon and DBG's herbarium. 76% of the cacti collections are in Grand Canyon and DBG's herbarium. So in doing any of the treatments for these families too, it would be really good to make sure they see our collections. So what do we see? So this is a Peach Springs cholla. It's a beautiful cholla in the west end of the Grand Canyon. It was first described from Peach Springs. And if you think collecting cacti are hard, try drawing them too. It's a real challenge. So one's only known from Peach Springs Canyon near the type locality. And because of trips with Wallapai Nation, because of park trips, and with our Arctomicon research too, we've expanded the range significantly from about river mile 169 to river mile 280. This bear grass, Nolina microcarpa, or is it Nolina microcarpa, has bugged me for years. And this is one we want to do some really good molecular, do the whole research with, and figuring out if it's a cryptic taxa. And you can see on the right, that's what we call Nolina microcarpa outside the Grand Canyon. So something's going on there. And if you look at SignNet, the map here, these are clues. When you see, and this is a small disjunct between Grand Canyon and the rest of them, but especially when involving an area like Grand Canyon, you want to look further into those species and see if indeed there's something different going on. People going down the Phantom Ranch, they walk right by this one. This is a prickly pear. It is definitely related to the brown spine prickly pear, Punchafea acantha, but I think it's also a new taxon, possibly a new subspecies. And the yuccas. I love yuccas, but in the canyon, they're very confusing. As a matter of fact, I sent this these pictures to Karen Clary, who's an world expert on yuccas, and she said, OMG, I have no idea. So this is up in Marble Canyon. We are doing molecular work on this one. Travel downriver in the central and western Grand Canyon. You got another one that we cannot put a name on. And I think there's 2 cryptic taxa right here. And then you have Yucca baccata. Everybody knows Yucca baccata. That's the banana yucca that produces these fleshy fruits. And then you have Yucca utahensis, and they're the capsular fruited ones. So even people who worked on yucca said, nah, yuccas don't hybridize. Well, these hybridize and form this, and it forms these fruits. It's like fruits don't know quite what it wants to be. And you have hybrids between 2 very dissimilar yuccas. And what makes it interesting too is Yucca baccata is a CAM plant, CAM photosynthesizing plant, and utahensis is a C3. And so they're hybridizing. And we actually have a gal that's looking into that, the CAM aspect. Go upriver in the eastern Grand Canyon, same thing, yeah, Yucca baccata. But here's this capsular one that we're not sure what it is. It could be a new cryptic taxa hybridizing with baccata, forming this hybrid, and it's not uncommon either. So this is, of course, an example of sympatric speciation through hybridization. Will they evolve into new taxa? Who knows? It's possible. So agave phillipsiana, we call it the Grand Canyon agave. Actually, Rose Column first rediscovered it back in the 30s in Clear Creek. And then Art Phillips, who's done a lot of botanizing the canyon, rediscovered it in a different canyon, told me about it, went down, collected it, and they're always found, the populations are found near cultural sites. These are ancestral Puebloan cultural sites. Well, we described it as a new species, and we added it to our ever-growing list of what we call pre-contact domesticated agaves, the PCADs, we call them. So first, phillipsiana was found in the canyon, then we found it in Verde Valley, then we found it southwest of Prescott, and then we found it east of Phoenix in Tonto Basin. And so here we have a plant that was grown, domesticated, and grown by regional people here in this area and traded with different cultures and growing them in different regions, different climates. And I think this is really interesting. They did not evolve in Mesoamerica, which most people, people think most crops originated from, but they're homegrown. And so this identified our phillipsiana from the Grand Canyon really got this going, and identifying the Arizona borderlands region as an overlooked but important area of plant domestication. So unfortunately, these plant domesticates, unlike animal domesticates, they're not protected. They have very little protection. They're not protected by any Endangered Species Act. They cannot be assessed for IUCN red list assessments. We have to figure out ways to protect the plant. And so we're developing a proposal. Carrie Cannon, who is with the Wallapai Cultural Resources in Peach Springs, Vincent Diaz, who is also Wallapai and Washoe, and he was hired by Grand Canyon Tribal Affairs Assistant and working with us. And we're going to figure out a plan and how to survey for and document additional populations, grow additional plants so that we can augment the known populations and monitor and co-steward these known clones. And we want the tribes to be involved in helping, and that's what we're working out too, and as well as getting funding. So we're really excited about that. So, but it does beg the question, what other plants have been cultivated by indigenous cultures? This prickly pear that's in Phantom, and other side canyons, it was probably brought in by ancestral Puebloan people, not only for the fruits, but also for the mucilage and used in their mortar. And that needs to be looked at as well. So we want to know, what are the processes driving speciation in such a unique area? And this is where it gets interesting. So have to look at how and when was the Grand Canyon formed. And this is pretty controversial. There's always new ideas all the time because it's very complicated. And a lot of the evidence got eroded away. So certain things we do know. From about 8 to 4 million years ago, during the Miocene, there was developed a desert environment. It was called Mojave. And that coincided with the uplift of the Rocky Mountains. And from 62 million years ago, it was beginning of an integrated lower Colorado River. And that was from about Hoover Dam to Yuma. Okay, so you see. So we're from here all the way down into the Gulf of California. At least parts of the Colorado River and Grand Canyon must be younger than 6 million years. Everybody can pretty much agree on that. So, so from 9 to 2 million years ago, you have the Bowser Mammoth, and then you have the continuous Colorado River flowing from Grand Wash Cliffs to the Gulf. So could there be isolation and speciation by the Colorado River and this what's called the Bowser embayment? The Salton Sea is a remnant of the Bowser embayment. This is influx of a lot of water. Could they have influenced divergence, isolation, divergence, and speciation? And it's called the riverine barrier hypothesis, not to be confused with the leaky barrier hypothesis. So Chris Smith and others said, yes, indeed, that these waterways were major factors in the isolation, divergence, and speciation of the western Joshua tree, Yucca brevifolia, and the eastern Joshua tree, Yucca yageriana. Not only the isolate and divergent the plants, but also their pollinators. Two distinct species of pollinators, Tegeticula antiphysalitica and Tegeticula syphilitica. And with animals, too. The Colorado River and Bowser embayment was believed to be important factors in the speciation, the separation and speciation of the Mojave desert tortoise and the Sonoran desert tortoise. And of course, this is an example of allopatric speciation by vicariance. And there are many other animal examples too. So then, okay, how about the Pleistocene? How did, if it did, the glacial, interglacial periods of the Pleistocene, which is about 2.6 million years ago to 11,000 years ago, affect Grand Canyon and her plants? We know that during the Pleistocene, there is a series, probably 20 to 25 of these glacial, interglacial cycles during the Pleistocene. And during these interglacials, especially with runoff from the Rocky Mountains, we have the warming climate, you got tremendous deepening, this is tremendous runoff. And so that is where you get the incising of the canyon. We're talking, they think, 1 million CFS, cubic feet per second. As a comparison, today averages about 8,000 to 12,000 CFS. So some believe that nearly half was carved during the Pleistocene, and most was carved by 1.2 million years ago. I have seen other reference that says, no, most of about 4 million years ago. So give or take a few million years. Also, there was many lakes formed down around Lava Falls, River Mile 174, right around there. That's an area with active volcanism. And so they would, in the last million years, especially in the last 250 to 500,000 years ago, a lot of volcanic activity, lava flows into the canyon. It would plug up, it would make dams. And there were many dams, some lasted tens of years, some hundreds of years before it broke down. One was so big and the backup was so long, they think it reached to the present town of Moab, Utah. That's as big a lake that is. So how the effects of lakes have on plant distribution and speciation. Did this corridor, was it a complete or a leaky barrier to plant gene flow, or did it encourage gene flow? Or probably it was both. And of course, how climate played in providing new niches, new habitats. Some are not going to do well, others are going to either adapt and possibly evolve in new taxa. So Lucas Major and others did this really neat study. It focuses on the dry-fruited group of prickly pears, genus Opuntia, and these are some that you might be familiar with: the beaver tail prickly pear complex, Basilaris, and the plains prickly pear complex, Polyacantha. So a little background, they think the prickly pears, the genus, arose about 7.5 million years ago from South America. Some stayed, others with northward migration. 6 different groups of clades moved north, originating in the Chihuahuan and Colorado Plateau regions. And this included this basilaris clade. There's further migration to the Colorado Plateau, and then others went elsewhere, the Chihuahuan Desert in Baja California. So this xerocarpa clade, that believed to have a Colorado Plateau origin about 5.2 to 2.1 million years ago. And this formed your basilaris group and your polyacantha group. So look at all these dots here. We'll try to make sense of all these. So these basilaris clade, we think, originated along the upper reaches of the Grand Canyon region about 2.9 million years ago, and then they spread south and diverged, and we have Elzada clovers and Lois Jotter's species, Longiareolata, the Grand Canyon beavertail, having evolved, but it is restricted to the upper half, the east half of the canyon. And then greatly influenced by the canyon, it spread further south and west to the Mogollon Rim and the Mojave and Sonoran deserts, and a little bit in the coastal range. Most are diploids. But here's the thing, though, too. They are adapted to warm climates, except Basilaris hylei up here. That is the least warm adapted species. And as you go farther down, evolve into the Longiareolata, that's a little bit more adapted to warm climates. And then, of course, your whole basilaris, your common beavertail, it grows in really hot environments. So in this paper, it was suggested that the Grand Canyon is actually a refuge for variety Longiareolata. When you look at the polyacantha group, this too originated in the Grand Canyon region a little later, 2.3 million years ago, very successful for various reasons, spreading into Chihuahua and the Mojave Desert regions, and a lot of them are polyploids. They're very well adapted to colder regions. My second example here, I'm talking about how the canyon can influence species, is one of my favorite plants, Newberry's mock yucca, Hesperio yucca newberryi. First, you had to prove, it's been proven that they are not a yucca. They belong in their own genus, Hesperio yucca. And today we recognize 3 different species. Newberryi, and you see it in the Grand Canyon. Note that it has only 1 stem. It doesn't produce more than 1 stem. Whipplei is, if you drive like to Los Angeles or San Diego, you're going to see them. And they produce 1 stem or many stems on a single plant. And then further south is Peninsularis, also produces either 1 or many stems. Well, there wasn't a lot of specimens of Hesperio yucca newberryi for quite a while, and for good reason. This is the kind of habitat it likes to grow in. So you got to be either really brave or really stupid to go after these. And Carrie, bless her heart, Carrie Cannon goes with me and helps get me these yuccas, Hesperio yuccas. And so we've built up the collection. But note this one right here. This is south of Arizona, right near the Arizona-Sonora border. Richard Felger collected this in 1984, and we went back down there a few years ago to see it. And we found it, and we documented with herbaриum specimens and did molecular work on it. And it is not Whipplei, which it had been called, but it is Newberryi. And so that is quite a disjunct with that species, right? Well, let's figure this out. So, like Joshua trees, did the balsam abatement in the Colorado River cause initial isolation? We don't know. And then another question too, did further isolation result during the Pleistocene? This is where it gets interesting. So during the Pleistocene, you have these glacial, interglacial periods. So when it's cooling, Everybody's happy. They're exchanging genes. There's no fragmentation. I'm pretty sure Newberii had evolved at that time, but we don't know. But they're doing great. Well, and then during the interglacials warming, different precipitation regimes, and there's fragmentation. Plants are dying, fragmentation of population. So this is going on 20, 25 times during the Pleistocene. And so at the end of the Pleistocene, about 11,700 years ago to the present, there was a rapid warming and drying trend. And all those little brown boxes represent pack rat mittens made by this little cutie, who they like to go out within 45 meters and collect just about everything. And then they bring them to their home, their nest, and they pee on them, and then it hardens them. And so researchers, thousands of years later, can figure out what is in that, what did that little pack rat gather, and date them. And so we know that Hesperonychus yucca, what found, did occur along the little Colorado, along the Colorado, corridor, but they're not there anymore. And this area is of interest because even during the glacial periods of the Pleistocene, it was warmer along here compared to the surrounding areas. So they had died out at least the last 10 to 12,000 years ago. So this is what we have today, right? But what about this guy right down here? Well, let's go back. So people inhabited along the river. We know people inhabited the area at least going back to 12,000 years ago. And they probably inhabited this area because there's water and the plants were there. And this was a theory that Richard Felger had suggested, that possibly they could have exploited the plants for food, similarly to how people use agaves, where they would bake the heart. So you use the whole stem. And if it's Newberii, the whole plant is gone. So that could have exacerbated the situation with the Newberii that are found along this region here. And we know that people have baked agaves for at least 10,000 years. So that makes sense. So, but what about this region right here? Richard Felger thinks that this area is so dry and so hot, no one would live there. And so it spared the populations that are there. And it's very, very possible that that could be that could have happened. Now, with the pollinators of Hesperonychus yucca, it's pollinated, all of them are pollinated by one species, Tetraticula maculata, and the subspecies maculata pollinates the California whipplei, and Extraña pollinates Southern California Baja whipplei as well as Peninsularis. So one of the few studies ever done with Newberii showed how, these pollinators are different. We think they're diverging, but they have the closest affinities with Extraña. And probably because of that bridge, or that Colorado River corridor where they used to be, that probably was the bridge towards the pollinator bridge with Hesperonychus yucca Newberii. And so they figured that Grand Canyon population was isolated from other Extraña for at least 11,000 years, and that the moth and plant's development distribution is related to these Pleistocene, Holocene, glacial, interglacial activities. So we noted before, exposure is really an important factor influencing plant and insect occurrences in Grand Canyon. So on one of our cultural trips, this is really exhausting work. It's to look and see if we can count how many Hesperonychus yuccas we can see on both sides of the river. Well, remember too, when we're going down the river, this is from the exact same spot on the river. You turn left and you see the north-facing slopes, you turn right, and this is your south-southwest facing slopes. And it does have a huge impact, we think. You know, this is a scientific methodology, but from the boat and counting, we were very diligent in counting. Hesperonychus yucca was 2 to 3 times greater on northwest, northeast facing slopes. And by the way, the Sonoran population, that one remaining one in Sonora, they were all north facing slopes. So the question is, how will Hesperonychus yucca do in the future? especially with climate change. There are so many studies done on Whipplei, the chaparral yucca, which grows in a more mesic environment, far more studies than for Newberrii. And I think one of the reasons is a lot of people thought that Newberrii was this conspecific with Whipplei. And since it's much harder to get to the Grand Canyon populations, number one, not only with difficulty in the landscape, but also you got to get permits from Wallapai Nation, from a national monument, and national park too. So they probably said, "Yeah, we don't need that. It's probably the same thing anyways, and we'll just focus on the easier to get to plants." And so, but they are distinct. They are two different species. So very few studies. We know hardly anything about them. So not only the plants, but also the pollinators. How are the pollinators doing? They spend a lot of their time as larvae in the ground. What triggers them to come out and do their cycle? And will climate change affect those as well? There's a cool program. It's Local Touchstones, Phenology and Climate Change that Grand Canyon does. It's mostly volunteers. And they selected like 6 different individuals of Gamble's oak. And so what they do is every 2 weeks, they note like when the leaf buds open, or when it starts flowering, or when it starts fruiting, all these life cycle aspects, and they record them. So I was thinking, that'd be pretty cool. We could do something like that with Hesperoreuca newberrii. And then I realized, okay, so I'm looking at it's 2025, and there's Carrie, 2026, not much going on. 2027, not a lot going on. And then like 20 years later, flowers. And so then, 3 months later, we got some good information, but it's like 20 years later. And so I realized, no, this isn't going to work. So we're talking with Carrie, we're coming up, we want to do a proposal that involves and is driven by Wallapai Nation, while I'm still alive too, to get this data on acquiring just the most basic information, mapping in a certain area and then modeling distribution, age classes. Why is it that there's like one plant flowering and you have to go another quarter of a mile to see another plant flowering? We want to do physiology, phenology data. This is just really just any kind of data would be new data for the species. What are the pollinators? Is it only the Tegeticula moth? And how effective are they? And when, there's hundreds of flowers produced on a stalk, and they could be pollen saturated, producing very few fruits, but too many times over the years, they're not producing any seed whatsoever. So there's so many questions we can ask. And so part of answering these questions is to get an idea of how what the climate is going to be, what they're projecting. And this report just came out this year. And so they have it under 2 climate scenarios. One's a warm, wet, and the other's a hot, dry scenario through 2050. And this is for Grand Canyon region, Grand Canyon National Park. So with temperature, it's what you'd expect, increase the temperature, of course, more so in the hot, dry scenario. And of course, more days added above 101 degrees, again, more for the hot, dry. But this is what really hit me. Historically hot years for wet, warm will be average temperature years in the future, but for hot, dry, average in the future would exceed anything experienced in recent history. For precipitation, and there's both scenarios, they both increase, but the amount, of course, is different. But with both too, you have increased extremes and intensity. And with drought, with warm, wet, it's less severe, but shorter drought-free intervals. In other words, longer drought intervals. And of course, with hot, dry, increasingly severe with shorter drought-free intervals. So I'm thinking about how this affects the plant communities in the canyon, and I thought of the riparian areas that have so much diversity in plant and animal life, how this will affect these areas. They're adapted to flooding and all, but if they're more intense and there's more longer drought intervals, how they will do. And it also points the question too of keeping an eye on introduced species as well. So, and one thing too I want to say, so in these, what's important with these side canyons and drainages, they can be refugia. It's a refuge for more cooler adapted species. For example, all these western redbug and beargrass, the Flavarium abduglii. So Grand Canyon is an important refugia for plants, and as importantly, if not more, an important reservoir for genetic diversity. Now, canyon can act as a refuge in this way, too. This is satintail, Imperata brevifolia. All those red dots outside the canyon, many of them don't exist. The plants don't exist there anymore because of habitat loss, groundwater pumping and all. But you can see Grand Canyon is this safe place for these plants. Same with Cladium californicum, California sawgrass. Again, same pattern. A lot of those are not there anymore. When you have the development of Glen Canyon Dam in 63, 64, it flooded a lot of populations. And so again, Grand Canyon is this refuge for this species. So future work, as with any work, more documentation, more sampling, right? We want to tease out possible new taxa. We don't want them to slip through the cracks and lose them. And this is something I got more interested in, is focus on other groups with similar distributions as our prickly pears and the Hespero yucca. And remember that Menzieli, the stickle leaves, they had such a different, one was in the Eastern Canyon and one was in the Western Canyon. So look at those and to test for this Grand Canyon signal. Maybe the canyon was very important in the evolution of these species, these and other species, in their biogeographic history. One of the things too is we've got good collaboration. We want to continue to build on that. We continue working with Wallapai Nation, get to go with their annual cultural river trips. And part of that is to visit and read transects that Art Phillips had set up in the early 1990s. And this is to see if there's any impacts that the dam had on useful plants for the Wallapai. So from that, we collect a lot of plants in the, for the herbarium, and Wallapai Nation has their own herbarium. So it's helping build up their own herbarium as well. And I want to mention this too. Peach Springs chapter, Wallapai have developed their own chapter for the Arizona Native Plant Society. It's called the Peach Springs chapter. And we think it's the only indigenous chapter for a Native Plant Society in the country. We're not sure of that, but we think so. So we love the collaboration. We also love our botany boot camps. Classroom one day, field work the next day. And we just had one a couple of months ago for the Grand Canyon Veg team. It's a lot of fun, and we really enjoy working with them. Grand Canyon Conservancy has provided us many opportunities to do service trips and going out and monitoring springs, collecting plants, building up the flora, really wonderful opportunities. And of course, our volunteers, I can't do it without them. And this too. So Lori was head of this idea. She was one that put this together into fruition. Was this wonderful exhibit. We held it in the Kolb studio twice. It was Grand Canyon's Green Heart, the Unsung Legacy of Plants. I would love to see that exhibit come back and exhibit and shown again. Everything about plants and botany in the Grand Canyon, through in the kitchen sink. And Lori, this is for you. We're so close to getting this. This was another of Lori's projects and ideas with Lisa Kersley, and working on and finishing up our Grand Canyon's rare, sensitive, and special plants with illustrations and photographs too. So in summary, Grand Canyon does have a high diversity of plant life due to several factors. It's really important to study and document these plants, understanding species divergence and potential discovery of new species. Grand Canyon is an important reservoir of plant genetic diversity, and it is a really important refuge for plants, as well as other organisms. We know so little, really. There's much to be discovered. what grows where. It's really critical to our being able to be stewards of this magnificent place. And it requires collaboration, and collaboration from many disciplines as well, and of course, involving Indigenous people. So I want to thank the DBG, and especially in Grand Canyon National Park. I've been working with the park for 30 plus years, and they've just been extremely supportive. Lori, so much so, Rhonda Newton and others that go back so long. Wallapai Nation, I've been working with them for 18 years now. Incredible experience, just wonderful people. Grand Canyon Conservancy, friends and volunteers. And finally, I want to thank Grand Canyon. Grand Canyon continues to give me humility, as well as always inspiring me. So with that, that is the end.